Hello, and welcome to our Spectronaut 15 launch seminar. This is Maximilian Helf from Biognosys, and I will be your moderator for today's event, which is titled Spectronaut Expand Biological Insights with DIA Proteomics. Here's an overview of the seminar, and as you can see, I will be joined today by Oliver Bernhardt, lead developer for Spectronaut, and Andreas-David Brunner, who is now a postdoctoral researcher at Matthias Mann's group at the Max Planck Institute of Biochemistry. At first, I will give you an introduction to our company and a short overview of Spectronaut. And after that, Oliver will introduce you to the new features in Spectronaut 15. And finally, Andreas will present on true single-cell proteomics, one cell at a time. And with that, let me briefly introduce our company. Biognosys has been pioneering new technology in proteomics ever since it was founded more than 13 years ago as a spin-off from Rudi Abersol's lab at ETH Zurich. And this has led to numerous patented innovations, and we are now operating one of the largest discovery proteomics facilities in the world. We are offering services and products to our customers around the world, and we keep expanding. And speaking of expanding, we're always looking for talented people to join us. So if you're interested in an exciting job opportunity at Biognosys, have a look at our careers page and visit biognosys.com for that. We offer three main software products for proteomics, and you will hear more today about Spectronaut, and that is our leading DIA Proteomics software. But I would also like to highlight that we have powerful software solutions for all proteomics applications, Spectrodie for targeted proteomics, and Spectromine for data-dependent acquisition, specifically isobaric labeling quantification. All our software is powered by a state-of-the-art database search engine, Pulsar, and Pulsar is also part of the reason why our software can provide very strong performance, leading identification numbers, and high speed across our entire software lineup. And our software is widely used in the scientific literature. And importantly, all software products are built with a strong focus on a user-friendly experience. But before we get started with Spectronaut specifically, let me briefly touch on the differences between data-dependent acquisition, or DDA, and data-independent acquisition, DIA. In DDA, MS2 spectra are acquired for specific precursor ions, and this has the advantage that ideally all fragments you see at MS2 level come from one specific peptide precursor ion, but this leads to a semi-stochastic sampling of the peptide population of a sample. And in DIA, on the other hand, a set of MS2 scans with a wide isolation window is acquired, and all ions in that wide range are fragmented together. This makes it possible to get MS2 fragmentation for all precursors in a sample across the entire retention time range and allows quantification at MS2 level. But as you can imagine, the MS2 data looks a lot more complex, and you need specialized software like Spectronaut to deconvolute these MS2 spectra and do quantification. Spectronaut is our best known software product and has emerged as the gold standard for DIA proteomics analysis. And with Spectronaut, you can generate spectral libraries and use them to help with your DIA analysis, or you can use our groundbreaking direct dia workflow that enables deep proteome coverage without the use of an additional library. So here's an outline of how direct dia works. And it's basically a two-step process. And in the first step, the DIA data is extracted into a collection of pseudo-MS2 spectra, which are then searched by Pulsar. The search results are then used to generate a spectral library, which then in turn is used for the targeted analysis of the DIA data. And unlike some other library-free approaches, direct dia also natively supports searching any post-translational modification. Direct dia can make proteomics a lot easier and can achieve proteome coverage comparable to using project-specific DDA libraries. And as you can see in this example, you can even break the 10,000 protein identification barrier when direct dia is combined with modern instrumentation. And please note that this is based on 2-hour gradients and only 6 biological samples without the use of a library. Deep learning can be used to predict physicochemical properties of peptides, and we make use of this at different stages of the pipeline, including for fragmentation and retention time prediction. And in Spectronaut, this deep learning augmentation is fully automated and is done without large impact on processing time. And with version 15, we improved the deep neural network to better handle non-triptic digests, and we can now predict eye mobility as well. And with that, I'm handing over now to Oliver Bernhardt, who will share with you a number of improvements that came with Spectronaut 15.
Okay, thank you very much. Thank you very much, Max, for the nice introduction. So I have the honor today to talk to you about what is new in Spectronaut 15. Obviously, there's a lot of stuff that we added and improved in the last year, so I had to limit myself to what I think are the most interesting aspects, and those are our improvements that we made to the general identification. So these are improvements for the classical library DIA search, but also for direct DIA, for the library-free DIA search. Our improvements in terms of quantification and quantification strategies in general. I also want to show off our new PTM analysis workflow today, and general improvements that we made to the user interface. There you will get only a small glimpse at it because we have more information about that in our recent YouTube video. But I will talk more about this once I'm there. So starting off with identifications. There, obviously, I want to compare Spectronaut 14 versus Spectronaut 15 to see where we have improved and by how much. And starting with the library-free analysis. So what you can see here in this plot is that we use Spectronaut 14 as a baseline. And we have a bunch of different benchmark experiments we use for this kind of analysis. And overall, you can see, so these benchmark experiments cover all kinds of gradients and methods and also different instruments. And overall, we improved about 7% on average on precursor identifications and about 5% on protein identifications compared to Spectronaut 14. But we explicitly excelled with improvements on short gradient dia-PASEF data, where we got between 25 and 30% improvements on precursor IDs, which is, in my opinion, quite noticeable. Similarly, we also improved our direct DIA analysis. There, a similar picture that we have about 10% improvement in identifications on precursor and 6% on protein level on average. But as you can see here, also we have a few huge outliers. Again, with dia-PASEF, where we just generally improved our processing for eye mobility scans, but also our four-dimensional feature detection, which leads to these nice improvements of up to 35% on precursor level for library-free searching of dia-PASEF data. As you might have noticed, that there are a few data points also that are below the Spectronaut 14 baseline here. And I want to go further into this because we have also made some changes on our false discovery rate control. And there we added some more flexibility for stricter criteria than was usually done in Spectronaut 14 before. So in Spectronaut 14, we only had run-level peptide FDR and global experiment-wide protein FDR. But with Spectronaut 15, we now also added run-level protein FDR, but also peptide posterior error probability control that you can utilize in your experiments. And I want to quickly go further into the difference between the FDR and posterior error probability, just for context. So those people who have used Spectronaut 14 in the past, or Spectronaut in the past, might know this plot. It's a Z-score histogram where that directly relates to the FDR calculation. And the way FDR is defined in this kind of circumstance is that you take the area of your identified port of the null set distribution that is above the cutoff line, and you calculate the ratio between this area and the total area of all peptides below above the cutoff line, and that is how you calculate the false discovery rate. In contrast to that, there is the posterior error probability that is not based on the area of the peptide distributions, but rather on the height of the peptide distributions. And why this is giving you a benefit, I will show in my next slide. So the motivation for this, for introducing the PEP, was that we've seen in certain experiments, especially with very highly specific libraries where you get an almost 100% recovery, that especially in knockout type experiments, it could happen that you see your knockout protein still in the experimental condition where it should be knocked out, essentially, and that is not an error in the false discovery rate control, but rather it is inherent to the FDR control with very specific libraries. And I want to further elaborate on that. Give me a second. So basically what this means is, if you think about it again on the slide I showed before, if you have a very a large library, you usually have a quite large set of peptides that cannot be identified, which is the one that is overlapping with the decoys here. And that is what is used for calculating the FDR. But if you get a library that is more and more specific, you diminish this null set, we call it, and this means that the false discovery rate cutoff moves basically to the left, because we can move it more to the left in order to even reach 1% FDR. And if you have a library that is completely specific, you basically move it so far to the left that you have everything included now, because even if you include everything, you still don't even reach the 1% FDR cutoff that you were aiming for. While on the other hand, the posterior error probability would still hover around this point here, so it would allow you to give more flexibility here on the identification control compared to if you only use the FDR control. And what we can see in practice now is that with this new filter options, we can effectively remove those false identifications from our knockout experiment and see exactly the on-off mechanic that we would have expected in this type of experiment. I want to next jump to the next topic, which would be the improvements we did to label-free quantification. Again, we are comparing Spectronaut 14 to Spectronaut 15 here. And here I want to talk again about, I want to introduce one of our controlled quantitative experiments. It is reminiscent of the LFQ sample setup, LFQ bench sample setup that some of you might know from the publication from 2015, except we used 4 different species that we mixed together in the ratios that you can see on the left. And we used a 120-minute gradient and the FAMES method and analyzed everything with direct DIA. The samples were measured in triplicates. And what we immediately saw once we analyzed both the data set, once with Spectronaut 14, once with Spectronaut 15, is that right off the bat, you immediately get about 20% more CVs on protein level below 10%. But you also, when you look at the regulation analysis and the known regulated proteins, we can see that here we also have a much better coverage of the true regulated proteins that are in the sample based on the known ground truth compared to Spectronaut 14, both of them using default settings. And the improvements are contributable to primarily the factors that you see on the side here, that we have improved normalization strategy. So we added a new automatic setting that will check the ideal normalization strategy based on the experiment you're using. We have introduced max LFQ as quantification option. We finally managed to have the time and resources to add this, one of the most requested features in the history of Spectronaut. What also contributes to these results is just the general improvements that we did to the DIA processing and direct DIA processing as well. But also the additional data filtering options you saw in the previous chapter obviously help in this kind of scenario. Moving on, I'm proud to introduce the new PTM workflow we have added to Spectronaut 15. And for that, I need to go one step back and talk about the history of PTM localization in Spectronaut. So last year, we finally got our publication published together with the group of Jesper Olsen in Nature Communications about our spin on the PTM localization in DIA. And it was a very successful paper and quite popular. And even this year, we had just three citations from Nature Communications pretty much back to back within the span of a few weeks, which made us obviously very proud. But one thing we realized is that a lot of people still need to do a lot of post-processing after Spectronaut in order to deal with PTM data. And the primary reason for that was because we didn't have proper site-level summarization in Spectronaut 13 and 14. So while we had very good localization of whether a modification was there, we didn't really provide the option to easily export site-level information out of Spectronaut. And that we improved now with Spectronaut 15 with our PTM workflow. So those who are familiar with Spectronaut know the report perspective, and there you have now a completely new type of report, which is called the PTM site report, where you have all the most important report options for a typical PTM workflow, like the flanking region around the site amino acid, which is quite useful for motif analysis. Or you have the protein, the site location within the parent protein, but also you have PTM site quantity now that you can use for downstream analysis. We also highly improved our protein coverage plot, which now features annotation for the localized and identified PTMs in the protein sequence, and with some interactivity as well, that if you click on the peptide coverage bars here in this plot, you get a preview of the XICs for the peptides that cover this particular protein sequence, essentially. Also with the possibility of reintegrating peaks and everything. So it's a nice new way of looking at PTM data directly in Spectronaut. Further, we have added a new post-analysis process for regulation analysis directly on PTM sites. You not only have the option to do regulation analysis on protein groups now, but also on PTM site quantities, which introduces an entirely new node in the post-analysis perspective, similar to the regular regulation analysis. But it also comes with additional principal component analysis plot, as well as correlation plot between protein fold change and PTM site fold change, so you can easily spot site modifications that do not correlate with, that do not correspond with the parent protein fold change, for example, which is a nice addition in my opinion. That concludes the PTM workflow slide, and I'm now happy to go to the next part, which is the improved user experience. And there we just cover basically all kinds of improvements we made to the user interface and generally raw data browsing. We made some cool improvements to our analysis perspective, actually a lot of improvements we added, but one of the most obvious ones for those who have used Spectronaut in the past would be our alternative data browsing option we have now in Spectronaut 15 in the analysis perspective. So we used to only offer the tree-like visualization that some of you also might know from tools like Skyline, for example, which also prominently features this kind of data browsing. But we also added now a completely new way of looking at your data in Spectronaut, which is the grid view, where you see more in a protein cross-run centric way. You see all the quantities, the log2 protein quantities of your samples. You have your sample columns annotated on top here, and we have again nice interactivity with protein coverage plots for all your proteins, and with XIC highlighting for the peptides of these individual proteins that you're looking at. Further, for all the people who do work with dia-PASEF and the timsTOF Pro, we added the ion mobility XIC, so the mobilogram XIC option for data browsing, including peak integration. And as you can see in this nice animated slide here, that you once you integrate your peak, refine your peak in the ion mobility dimension, you can see that the peak in the retention time dimension, which is directly dependent on it, automatically updates, and so you can better control for explicit candidates. You can now really fine tweak your peak integration if you want. We obviously added a lot of other features as well to Spectronaut 15 that are too numerous to talk about here. But just as a quick summary list, we have improved our analysis log. We added the quick action bar with the most important action buttons, basically for the analysis perspective. We added detachable perspectives and floating plot windows, and overall better navigation functionality for cross-run plots and just how to navigate in the tree view and the grid view. But all of this is obviously a bit difficult to show in a slideshow, so I would again highlight the YouTube video of my colleague Fabio Simona, who goes nicely into all of these new features in a way more visual way that I ever could here in this webinar. So I would recommend you go to YouTube and look for our YouTube channel Biognosys and look at the YouTube video there. Or obviously you can also just click on the resources. I always keep forgetting that on the side you should have this resource tab, and it should be linked there as well. This already brings me to my summary slide. So obviously Spectronaut 15 is for a better DIA analysis. We always strive to be better than last year, but specifically what makes Spectronaut 15 provide better solutions for you is the increased identifications, better quantification, the better filtering options you have, our new PTM workflow, and obviously just overall better data browsing using the improved user interface of Spectronaut 15. Thank you very much for your attention, and I hand back to my colleague Max. Thank you.
Yes, thank you very much, Ollie. I think that gave a very good overview of what's new in Spectronaut 15. And please, if you have any questions about Oliver's talk, you can send them now in the Ask a Question box. And yeah, like Ollie said, resources are available on this platform as well, like the videos that he mentioned. And now it's my pleasure to introduce Andreas-David Brunner. He recently finished his PhD in the Matthias Mann Lab at the Max Planck Institute of Biochemistry. He has had a very successful time as a PhD student and has published a lot. So yeah, I think he can be really, really proud of his work. And we are looking forward to hearing about true single-cell proteomics that he has worked on over the last few years. And yeah, I think with that, Andreas, please, yeah, the floor is yours. Thank you.
Hello. Yeah, thank you very much for these kind words and for this nice introduction. Yeah, first of all, I want to say thank you very much to the Biognosys organizing team for giving me the opportunity to present our latest work. And yeah, so I'm Andreas Brunner, by now a postdoc in the lab of Matthias Mann at the MPI of Biochemistry Munich. And I want to talk about our way of performing scalable and quantitative single-cell proteomics by injecting one cell at a time into the mass spectrometer. So first I want to answer the question why we actually need single-cell proteome analysis. So in any omics technology, and especially in proteomics, we aim for the measurement of any given sample in the deepest and most quantitative way as possible. And this in many cases comes hand in hand with the need for very large initial sample amounts. And for example, in the analysis of tumor samples, we often obtain so-called tumor thin sections, which we then scrape off from a glass slide and process it as a whole to obtain, for example, a very deep proteome of this particular tumor sample. The issue here is that we will inevitably obtain a merged proteomic signature of often highly heterogeneous tumors that are either filtrated or surrounded by, for example, immune cells, which when analyzed separately could yield very, very valuable information about how tumors and their microenvironments work, and would also point towards a possible therapeutic intervention. So this is why there's an unmet need for single-cell proteomics technologies or analysis in general. So the RNA sequencing community did a very good job throughout the years in developing new technologies or methodologies for sequencing of single cells, and also especially computational approaches to handle these large amounts of data. And historically speaking, it started only 12 years ago in 2009, when the first paper on single-cell RNA sequencing was published, where they sequenced only a handful of cells. So the field then had its major breakthroughs in 2012 with the advent of SmartSeq, and in 2015 with the development of the DropSeq technologies, which are currently dominating the market as the very famous and well-known 10x Genomics platform. And this, of course, resulted in many publications, but especially recently in publications which contain several millions of single-cell transcriptomes. So proteomics itself is unfortunately lagging behind quite a lot in this field, which is mainly due to the classical workflow you have to apply to obtain a proteome. And there are, traditionally speaking, three pillars, which are the sample preparation, the liquid chromatography coupled to mass spectrometry, and then also the spectral interpretation and the downstream analysis. And all of those work very well and super robust for large sample amounts, but they definitely have their major drawbacks for ultra low sample amounts. And our goal in this study was to adjust or reinvent all of those pillars to enable single-cell proteomics. And we started most intuitively with boosting the sensitivity of our mass spectrometer. So our mass spectrometer of choice is a modern quadrupole time-of-flight mass spectrometer, which possesses a trapped ion mobility device right at the beginning of the ion path. And the difference to a traditional timsTOF here is the dual TIMS analyzer. So ions are dragged into the mass spectrometer by a carrier gas, where they face an opposed electrical field gradient with increasing strength within the TIMS device. And this leads to the separation of the ions by their ion mobility or their collisional cross-section. And ions with a higher collisional cross-section and lower ion mobility make it further into the gradient. And ions with a smaller collisional cross-section and higher ion mobility will be trapped earlier in the TIMS tunnel. And this first results in the distribution of the interfering chemical noise, as you can see to the right, yielding virtually noise-free mass spectra. And second, the peak capacity is increased by more than tenfold by the means of ion mobility resolution. And third, this principle leads to very, very dense ion packages of the ions of interest, which you then also want to analyze. So this ends up in a very high signal-to-noise ratio. So the dual TIMS analyzer also allows us to adjust the voltages independently within the TIMS device, so that we can then scan the ions in very unique ways, either for data-dependent or also for data-independent acquisition scan modes. So the DDA scan mode, which is unique to this device, is called parallel accumulation followed by serial fragmentation, or short PASEF. It is divided into 4 steps, and I will start to explain it from the bottom of this slide. So in the first step, ions are accumulated in the TIMS tunnel 1, and this happens independently of the TIMS tunnel 2. And then this particular ion package is released into the second TIMS tunnel and separated by its ion mobility, while other ions of the incoming ion beam are accumulated again in the TIMS tunnel 1. In a third step, these ions are separated then by their ion mobility in the TIMS tunnel 2 and slowly eluted from this device. The trick is now to synchronize the quadrupole isolation window with the elution profile of the ions from each TIMS scan in a serial ion fragmentation fashion. And this then also we adjust the collision energy which we apply to each of these isolation windows based on the ion mobility values for fragmentation. And this ultimately allows us to sequence more than 10 precursors per TIMS ramp and up to 100% duty cycle, which increases naturally sequencing speed and sensitivity by definition by up to 10-fold overall. So with this setup at hand, our first goal towards single-cell proteomics applications was to benchmark the sensitivity limitations of our machines back then. And to do so, we acquired data for a HeLa digest dilution series starting from 25 nanogram, going down to 0.8 nanogram of starting material. And this approximately corresponds to 3 pooled single cells, when keeping in mind that a single cell has approximately a protein amount of 150 picogram. So surprisingly, we were able to already quantify more than 550 protein groups with a very high qualitative reproducibility. Also, the quantification behaved fully linear across the dilution series, and even the 0.8 nanogram replicase correlated very well, hinting that we should be able to quantify very low sample amounts. So unfortunately, at this point, we also had to realize that we were hardware-wise not yet at the level at the sensitivity and ion transmission efficiency level that we needed to perform true bottom-up single-cell proteomics, in which we process one cell at a time and inject one cell at a time into the mass spectrometer and purely identify and quantify peptides that reside from single cells. So we set ourselves the challenge and wanted to develop a workflow and technological setup to reach the sensitivity and robustness needed to quantify thousands of proteins from hundreds of cells at highest quantitative accuracy when we inject one cell at a time into the mass spectrometer. So this meant for us that we had to increase the sensitivity of our full setup by at least 10 to 50-fold at highest robustness to maintain sensitivity over thousands of runs. So together with Bruker, this led us to develop a modified version of our current mass spectrometer with much increased overall ion transmission and sensitivity with regards to the whole ion path, starting from the ion source and optimized ion routing. So next we evaluated the raw intensity increase for a 10 nanogram tryptychila digest and recognized a more than 4-fold increase of intensity on the MS1 feature level for shared peptides only, compared to our standard instruments. And at this point, we decided that it was time to test our setup for the analysis of real single cells. So therefore, we FAC sorted single HeLa cells with an increasing cell count from 0 to 1 up to 6 cells into 384 well plates, processed them with our workflow at below 2 microliters of total volume, and analyzed them with our modified mass spectrometer tuned to maximum sensitivity and ion transmission. So we were able to quantify close to 1,000 protein groups from single-cell replicates, controlling the FDR at all levels at 1%. So also, we could see that the numbers of protein and peptide identifications increased linearly with increasing cell count. So highlighted in white and gray at the 3 cell count measurements, one can see that those are most likely failed measurements that happened because of the failed cell sorting process, issues during sample preparation or sample loading, which is one would also expect working with these low sample amounts, and truly reflects the experimental challenges to perform these experiments. So interestingly, when then comparing isotope patterns of single and 6 cells, we can also for any peptide of interest, we could appreciate a 6-fold increase in intensity levels. And this really hints that we can quantify peptides even at these low levels correctly. So at this point, we were running, unfortunately, into performance decay issues that were related to the chromatography setup. And since we are using more and more a novel chromatography platform, which is called the EvoSep1 in our laboratory, which allows proteome analysis at highest robustness and the measurements of, for example, thousands of pull downs in human and yeast interactome studies, we decided to also give it a try for single-cell proteomics analysis. And in this LC, a peptide-loaded disposable trap column is moved into the LC system. Peptides are then eluted into a preformed gradient and finally pushed out under the control of a single high-pressure pump. So being able to push out these peptides with the preformed gradient has indeed many advantages. Like we can control the flow rate of eluting peptides precisely, and we reason that one could even control flow rates in the very low nanoliter region with this strategy. Also, the disposable trap column which one could then load single cells for processing, has many advantages, like reducing carryover close to zero between the runs, concentrating the sample into very low volumes, and a simultaneous peptide cleanup, which would then also allow us to use, for example, salt-based protein solubilizing agents. So interestingly, this idea allowed us to control and scale down the flow rate within our measurements as we wished. And we could then even keep the pressure and flow rate stable at 25 nanoliters per minute, as shown to the right. So when we then also compared the analysis of only 1 nanogram of HeLa digest between the standard microflow gradient, which is also offered by the EvoSep, and the 100 nanoliter flow rate version of this particular setup, highlighted a signal increase of more than tenfold, which is one would also expect, since electrospray efficiency is also highly flow rate dependent. So furthermore, standardizing the setup to 100 nanoliter per minute gradient finally enabled us to run more than 1,500 runs or measurements with only a marginal increase in back pressure, and most importantly, without any performance decay. So one of the most important and sort of limiting steps in the process of performing single-cell proteomics is literally to find a way how to transfer the only about 150 picograms of peptides from a single cell into the mass spectrometer. And we solved this issue by combining the sample preparation workflow with the liquid chromatography system. So here we sort single cells into a 384 well format, process these cells in a 2 microliter total volume, transfer them into the EvoTip, and concentrate them. And this is done in a 96 well format, which enables parallel processing of these single cells at scale. Then the LC system picks one tip after the other and elutes these peptides into the mass spectrometer, controlled by a single pump running at 100 nanoliters per minute, with a throughput of approximately 40 single cell measurements per day. So one very important, if not game-changing aspect here is the peptide concentration effect. And this results in a very small peptide nanopackage of the single cell peptides at the interface of the EvoTip. So also the elution of this 20 nanoliter nanopackage happens in a sub 100 nanoliter volume, which then also ensures that the peptides of single cells are not diluted at all in the LC system and are positioned right behind at the analytical column. So with this at hand, we now also had to find a way how to make use of this increased and incoming ion beam into the mass spectrometer. So we also developed a novel data-independent scan mode, which is based on this PASEF principle and uses the full correlation of the M/Z to the ion mobility in the TIMS device. So here the quadrupole is fully synchronized with the elution profile of the ions from the ion mobility ramp, and this allows up to 100% ion utilization, compared to sometimes even less than 3% in standard DIA applications. So combining all these developments and optimizing the scanning even further, we were able to identify more than 3,200 protein groups from only 1 nanogram of tryptic HeLa digest. And compared to the start performance of the technology back then, this increased identifications by more than 600%. So more interestingly, when using the latest version of Spectronaut, which is Spectronaut 15, even though it controls the FDR now even on more layers more stringently compared to the 14 version, we observe an increase in identifications to more than 3,500 protein groups, which is a more than 10% gain in protein identifications. So now it was time for the real large-scale single-cell experiment, and we asked if it is possible to dissect the cell cycle on the single-cell level by drug perturbation. So to do so, we treated HeLa cells in culture with either nocodazole to block them in the transition of the G2 to M phase, and also with thymidine to block them in the G1 to S phase transition. Then we remove these drugs as perturbation and let the cells grow either into the G2 phase or the G1 phase. So we ended up with a total of 4 conditions, of 4 cell cycle stages. So we sorted these 4 conditions, cells of these single cells of these 4 conditions, and subjected them to the described LCMS setup, tuned to highest sensitivity and robustness, and identified up to 1,300 protein groups per single cell using Spectronaut 14. So using the latest version of Spectronaut, so 15, boosted the identifications to close to 1,600 or even 1,700 protein identifications. And what was very important for us is that this really happens without inflating proteins from unexpected low abundance ranges. So next we asked if you can also pick up quantitative heterogeneity between the single cells. And PCA analysis of this experiment allowed us to separate the single cell measurements according to their cell cycle stages. So we also asked if we can predict, for example, the cell cycle stage from our experiments with published and very well-known marker proteins for any cell cycle stage comparisons, and rock curve analyses showed that the single cell measurements correlate well with the expected stages. So we also went for differential abundance testing between the G2M and the G1S phase single cell measurements to test for known cell cycle stage markers, which we covered many of them. So for example, we found very strong markers like STATMI1 to be downregulated in the G2M phase. We also found CDKN2A and many topoisomerases being downregulated in the G1S phase. And interestingly, we also found members of the ubiquitin pathway to be regulated in the correct phases and many more potentially novel proteins. So one very important part in performing these measurements, or in generally performing proteomics measurements, is the quality control. And so to quality control our differentially expressed proteins, we also set up a pipeline to visualize extracted ion chromatograms, fragment intensities, and BNY ion series for each of the matching precursor ions as shown exemplary here. We also inspected MS1 isotope envelopes, as well as MS2 fragmentation spectra, and also MS2 fragmentation intensity correlations for the identified peptides to check for their correctness. So interestingly, one can now also inspect the ion mobility extraction windows to add an additional dimension of quality check in the new Spectronaut 15 version, providing literally all possible layers of visual information that one can then use to really QC their data. And I personally like this a lot, and I'm applying this to literally all of my workflows. So with this single cell proteomics workflow at hand, we are now also able to ask very fundamental questions with regards to how this layer of information compares to other single cell layers like the transcriptome, which is often used as a proxy for protein expression levels in cell typing endeavors like the human cell atlas. So to do so, we compared cell type matched single cell RNA-seq data from 2 state-of-the-art single cell technologies, which are DropSeq and SmartSeq, to prevent bias to the single cell proteome data. And in a PCA, we were able to separate the proteome well from single cell RNA, from both single cell RNA-seq technologies. Also, when correlating all shared genes between both layers of information, highlighted that transcriptomes from both technologies correlate highly between each other, while they do not correlate well with the proteome data. And this again highlights that both levels of information are indeed very different, but yield very complementary information. So furthermore, when comparing the expression values of 2 single cell proteomes, it sort of struck me in a way because it showed a very high correlation to each other and highlights that single cells must have a stable proteome. So investigating the quantitative nature of these data across the whole drug-perturbed single-cell experiment by plotting the mean expression values for each protein against their coefficients of variation showed that the data do not follow a shot noise or negative binomial distribution, which is often used in single-cell RNA-seq data to model that data. Also, we reasoned that single cells can have or should have a set of proteins that can assemble a core proteome, which are essential for function and are not subject of drastic change, as highlighted in orange. So comparing these proteome data to the single-cell RNA-seq data revealed that indeed single-cell transcriptomes are rather volatile in their expression levels compared to single-cell proteomes by the means of coefficients of variation. And this is especially true in case of the defined core proteome as highlighted in orange. So this established single-cell proteomics workflow can, of course, not only be used for the analysis of single cells, but can virtually be applied to any sample-limited application. And in a fully cell-type resolved concept that we call deep visual proteomics, thin tissue sections are isolated, a deep learning model is trained to automatically identify single cells and order them by their cell type. So then it teaches the microscope to automatically isolate and pool these single cell types, followed by the analysis with the ultra-high sensitivity single-cell proteomics platform I just presented, followed by downstream analysis, which then could yield very valuable information for clinical as well as biomedical decision-making. And this approach already highlighted substantial cell type heterogeneity between tumor and non-tumor cell types, and also differences in the spatial proteomic makeup of tumors. It also showed, and this is to me super interesting to see, that the cellular phenotype is tightly connected to the proteome and opens up the possibility for cell type resolved spatial proteomics applications. So in summary, I showed you how TIMS benefits ultra-high sensitivity measurements, how we developed a novel LC-MS and sample preparation workflow enabling single-cell proteomics. Also, I showed you that we can pick up single-cell proteome heterogeneity upon drug perturbation, highlighted the single-cell proteomes are very different compared to transcriptomes, and closed with a very exciting application in digital pathology. So with this, I first of all want to say thank you very much to my doctor father Matthias Mann, who is enabling these breakthrough projects always in the best way possible. Also, thank you very much to all the people at Bruker, to all the people from our daughter group in Copenhagen, all people at Max Planck, our collaboration partners from EvoCep, the Helmholtz Center, and Biognosys, who are involved in making single-cell proteomics a reality. Thank you very much for your attention, and I'm happy to take questions.
Yeah, thank you very much, Andreas. It was a great talk about single-cell proteomics, and we will move on to the questions. We actually got quite a few. So first one is for Andreas. So for the single-cell proteomics, did you benchmark different cell types? Because cells are different in size, have different amounts of protein in them, I guess. So yeah, are there differences? Some cells better than others?
Yeah. So that's actually a very good question. So because one would expect this to happen, so we performed these experiments on HeLa cells simply because yeah, it's kind of the standard cell line in our laboratory that we use on a daily basis. So what we could see there is that, of course, one would expect that cells are growing during the cell cycle, starting from G1 where they are smallest, and then they grow. And there we actually see differences in raw signal. So the raw signal is increasing across the cell cycle stage, which correlates with the cell size and the total protein amount a cell offers. We also tested these measurements for other cell lines, and this also works very well. And yes, we are also currently looking into other applications for tissue proteomes. For example, where also cells vary in their size.
Okay, thank you. And we have a question here for Oliver. So is there any way in the software to calculate the percentage of occupancy of a specific PTM to understand if a specific phosphosite is, for instance, 100%, 50%, or 10% phosphorylated?
Yeah, very good and legitimate question. We had this on our radar. However, it's a bit more complicated because you basically need to compare two different experiments against each other. So a non-phosphor-enriched or in a phosphor-enriched experiment. And currently, the mechanic of combining two experiments in Spectronaut into one post-analysis is not there yet. But we do have it on our wish list for Spectronaut 16, and we hope we can work and improve the PTM workflow further in this direction.
Okay, thank you. And for Andreas, we have another question here. So what is the lowest number of cells needed for accurate or reliable quantification? This user gives an example for around 100 cells per condition.
It's one cell. So so it's literally one cell. I mean, like, so you are comparing one cell against the other, and I mean, like, so the more cells you have for, I mean, like, every cell is unique in its makeup, right? And this is also what we see. Of course, there are cells which are extremely similar. But there are also cells which, even within these, within the same cell cycle stage, that they are quite heterogeneous. And this was really cool to see, because this is what you would actually miss completely in a bulk experiment, right? So yeah, this could be an answer to this.
Mm-hmm. Thank you. I guess it's hard to hard to just get on board with single cell proteomics, right? Because it seems like such a big step. So I have a question here for Oliver. So is there any new features regarding batch variation correction in the new Spectronaut? And will you consider that in future releases?
Oh man, again, another one where I have to disappoint. No, currently we don't have batch correction in Spectronaut 15, primarily because there are a lot of different flavors of doing it, and we haven't really figured out yet we cannot implement every possible possibility. So we leave it right now up to the user to who knows his samples best to know how to perform batch correction. But it's also something that we have on our radar for future releases as well. Yeah, that would be the answer to that.
Okay, thank you. I have one for Andreas here. So what kind of samples, tissue types have you analyzed so far? Only FFPE tissue, or also, for instance, SNAP frozen tissue?
Yeah, so for now it's only so for the single cell workflow, we were only working on cell cultures, and we are moving slowly into fresh frozen tissues. It looks okay so far. For the deep visual proteomics workflow, we use FFPE tissue.
Okay, thank you. And here's a question for Oliver. So is PASEF more beneficial for direct DIA or for the library-based approach? And what differences in IDs can you observe Spectronaut 14 to 15?
Good question. So what you will definitely see is that the jump between Spectronaut 14 and Spectronaut 15 is bigger now with for direct DA, where we had more different aspects where we could improve the pipeline. For the library-based analysis, we also made significant improvements, not as drastic as for direct DA, but still in the range of 10 to 15% compared to Spectronaut 14. And in general, I forgot what I wanted to say now. So you definitely see improvements in both workflows, and especially with the new ion mobility prediction that we have to the library generation, where you can now use libraries that were acquired on different instruments that are not ion mobility capable. So you can expect an improvement for direct for timsTOF data of around 15 to 20% for library-based, and we saw consistently up to 30% for direct DIA in terms of improvements.
I guess there's another question that's related to this. So how do results of DIA analysis differ if you use this empirical eye mobility versus the in silico generated eye mobility?
I would have to think about that. Is we also see, so if it makes a huge difference if you don't have any eye mobility, obviously, annotation in your library. Obviously, the empirical is preferential. That one will always be more accurate, but it is a good. The predicted eye mobility is very much on par with the empirical eye mobility and gives you very similar results if you happen to have no access to library generation from a PASEF instrument. It's a good alternative, I would say.
And with that, I would like to thank our speakers, Oliver and Andreas, for your insights today. It's been a real pleasure having you with us. And also thanks to ASO Network and Insight Scientific for their support in the production of this webinar. And last but not least, we'd like to thank the audience for joining us today. And we hope that you enjoyed the webinar. Thanks again, everyone, for attending this webinar, and we'll see you again next time.